Hi, I'm Jeong, a postdoctoral research associate at Dr. Raphael Pinaud laboratory at the University of Rochester. We will show you double fluorescence in-situ hybridization in fresh frozen brain sections to detect two mRNA transcripts simultaneously at single-cell resolution. This method can help answer key questions in the neuroscience field, such as properties relating to the anatomical, functional, molecular, and neurochemical organization of vertebrate brain circuits.
Though this method can provide insights into gene co-regulations in neurons of the avian brain, it can also be applied to other systems, such as rodents, as well as a variety of non-neural tissues.
To begin this procedure, hydrate an adequate amount of Cephidex G50 powder with RNase-free water. Mix the solution briefly and store at room temperature for 2 to 5 minutes. This step separates the Cephidex beads from the dextrose found in commercial Cephidex powder. Following the Cephidex G50 precipitation, remove the supernatant and repeat the process 3 to 5 times. After the final wash, resuspend the Cephidex G50 solution in TE buffer at a 1:1 buffer to bead volume ratio and store at 4 degrees Celsius until use. Next, prepare the columns. Place autoclave glass wool into a sterile 1 ml syringe and compress it with the plunger to make a compact layer at the bottom. Then place the syringe in a 15 ml Falcon tube. Fill the columns with